A study was carried out in 1973 to determine the physical condition, parasite burden, and reproduction of black-tailed deer in Humboldt County in northwestern California. A permit was granted by the California Department of Fish and Game to wildlife researchers from Humboldt State University to collect 24 deer and additional roadkills. The deer were taken from 3 major habitats in this area: second-growth redwood forest, oak woodland and prairie mixture, and coastal brushland. The baseline information that was gathered revealed the current status of the herds and provided statistics with which to compare future changes. This film will show the procedures used in gathering the data results. This cool, moist, frequently overcast part of California, shown here near Big Lagoon, is known for its extensive stands of redwood forest, most of which is second growth and is mixed with Douglas fir, Sitka spruce, and red alder. Most of this coastal redwood forest is owned by large lumber companies which carry on a regular routine of logging. This provides newly logged areas as well as old cuts in various stages of regrowth. This situation of alternating open areas and areas of dense cover provides a nearly ideal situation for deer. Much of Humboldt County, inland from the coast, consists of a mixture of woodlands and meadows which are locally called prairies. The vegetation consists of open stands of Oregon white oak and California black oak, with a variety of grasses making up the understory. The water courses and ravines are overgrown with bay laurel or red alder. The 3 study areas were chosen with their diversity in mind in order to study the range of difference between the herds living in different habitats. This land is primarily used to raise beef cattle on scattered ranches, such as this one in the Neeland area east of Eureka. The coastal brushlands in the Kings Range National Conservation Area are steep, dry, and chaparral-like. Fewer tree and grass species exist, with meadow-like openings such as this one being rare. This habitat was the poorest in its ability to support a herd of deer. Much of the brush was only useful as cover, being utilized only as starvation subsistence, and favored species were heavily browsed and sometimes showed severe signs of overuse. Severe overbrowsing is shown by this wavy-leaved ceanothus, which has been cropped to the ground and reduced to a prickly mat. Stands of preferred foods, such as these whitethorn along deer trails, were eaten back as far as the deer could reach. On many plants, nothing was left but tough, almost inedible twigs and thorns. Scattered clearings and fire breaks provide openings and edges where deer were commonly seen and collected, and large stands of manzanita blanket extensive areas, providing some cover, but virtually no food for the deer. Road hunting was the typical method of collecting the animals. The vehicle was driven at 3 to 5 miles an hour until a deer was spotted within rifle range. Collections were restricted to adult does in order to get as much reproductive information as possible. 2 does were collected in February, May, September, and December from each of the 3 areas to see what the seasonal changes were within a herd, as well as the differences between the herds. The shot was always aimed at the neck to ensure either a clean miss or a certain kill. The time elapsed from the firing of the shot to death was recorded, as well as the exact location, the weather conditions, and air temperature. A portable field autopsy kit, in this case a large tackle box, contained all the necessary items for working on the deer and for taking sterile blood and tissue samples. Statistical data on the size of each deer were kept, such as girth measurement, which was taken before cutting open the body cavity. Other weight and length measures were taken later in the laboratory. Before the blood had a chance to settle or coagulate, a sample was taken directly from the heart. The sterile samples were refrigerated upon returning to the university, and at a later time, complete analysis done of them. In order to analyze the internal bacterial flora, tissue samples were taken from the liver, spleen, and lungs, and also feces. One of the pathogens looked for was Listeria monocytogenes, which can potentially reduce reproductive success by causing repeated spontaneous abortions, and it has been found in a nearby elk population. An examination was made in order to determine the degree of infestation by ectoparasites. One of the most common ones in all 3 study areas was the hippobossid fly Lipoptina, which in the adult stage is wingless. Ticks were equally common. This is a female dermacenter. When feeding, female ticks become greatly engorged and swell up to many times their normal size. This is the winter tick, Exodes pacificus. The incidence of eyeworm, Phalasia, was extremely low. This doe had none. Phalasia is a filarial nematode that is probably passed by flies. The external parasite burdens that the does carried usually cause no harm in low numbers. They are just part of a normal body fauna that the deer carry. Hippobossid flies and Exodes ticks were most common in winter in all three herds. Most ectoparasites were least common in September. After two animals were collected from an area, they were transported to the Wildlife Disease Facility at Humboldt State University. A variety of standard measurements were first taken to see if there were significant differences in size and weight from the three collection sites. The total body length was recorded. It was found that in terms of both length and weight, there seemed to be a general increase in size of the animals as they got older, up to about 6 to 7 years of age for the redwood and woodland prairie habitats. For the does from the overbrowsed coastal brushland, there was, on the average, no increase in size after 3 years of age. The so-called shoulder height is measured from the tip of the hoof, not to the shoulder, but to the middle of the back. A useful indicator of size when a wildlife worker does not have an entire carcass to work with is hind foot length, which correlates very well with general body size. The data were entered on a large data board so that they could readily be referred to and comparisons could easily be made. The body weight and length measurements revealed that the does from the redwood sites were usually heaviest and largest. Other measurements, such as depth of back fat, showed that the woodland prairie deer were the fattest, while the coastal brushland deer were the smallest and contained the least body fat. The feet were removed and set aside to be later checked for footworm, Werdikmansia cervipedis. This is a nematode parasite that lives in the connective tissue between the skin and muscles and is most commonly found on the lower feet, but in heavy infections spreads up as far as the back. Footworm is often encountered by hunters but does not harm the meat. This parasite is passed by a black fly which picks up footworm larvae when it takes a blood meal from the ear of an infected animal and passes it on by feeding on another deer. These worms are often 8 to 10 inches long and are part of the normal parasite fauna of deer, causing no harm to the animal. The reproductive success of each of the three areas varied. Does from both the redwood and the brushland habitats had an average of 2.0 eggs per doe, while those from the woodland prairie habitat had an ovulation rate of 1.5 eggs per doe. It was found that on the average of the eggs produced, 78% were subsequently fertilized. Possible reasons for this low value are an accident of small sample size, extracorpora lutea may be produced, as are done by some elk, or it may be a sign of reproductive problems for these deer. The ovaries were cut out and put in 10% formalin. Later, after a minimum of 24 hours to allow them to harden, they were sectioned and checked for ovarian structures. The number of corpora lutea in the ovaries reveal how many eggs were shed, or the current year's ovulation rate. By counting the number of corpora albicans, the brown corpora luteal scars, previous year's ovulation rate can be computed. A number of the does were pregnant. It was found that the breeding season for all three herds is very similar. The peak is in late November, but ranges from late October to late December. The average number of fawns born to each doe was lowest in the redwood area, 1.25 per doe. In the brushland, it was 1.4, and in the woodland prairie, it was 1.5 fetuses per doe. By comparing the ratio of fawns and does in an early fall herd composition count with the fetal rates, it was estimated that about 75% of the fawns produced were dead within a few months after birth. This mortality rate is higher than reported in other studies. After measuring the length and weight, standard tables were used to determine the age of the fetus, and this was backdated to determine the breeding season. The kidney fat index is used as a measure of general health in a variety of ungulates. Research with other deer herds in other western states has shown that it correlates well with the true condition of the deer. This procedure involves carefully cutting out the kidney along with any fat that may surround it. The excess fat is trimmed off from the top and bottom, and the capsule that encases the kidney, along with the fat that adheres directly to the capsule, if any, are skinned off. This kidney from a doe from the brushland had virtually no fat. The weight of the capsule and the fat divided by the weight of the kidney is what is termed the KFI. there was a great degree of seasonal variation in the redwood and woodland prairie herds. For comparison, this is the fat from a woodland prairie deer. The fat deposits in the bone marrow of a deer have also been used to determine general physical condition. Just as with kidney fat, animals in better condition lay down larger amounts of fat in the marrow. The amount of marrow fat was determined by a Soxhlet apparatus. Again, there was considerable seasonal variation. After sectioning the femur, a simple visual comparison can be made between the marrow of deer from different habitats. The one on the right, taken from the woodland prairie, is much lighter in color because of the fat it contains. It is also greasier in appearance and touch than the sample on the left, taken from the brushland, which is much redder because of the absence of fat. These condition studies, along with the blood value analyses, suggest that the deer of the three study areas are on different annual cycles. For the redwood deer, the peak time of the year is the summer and fall, while the late winter is the poorest time of the year. The winter is the best season for the woodland prairie deer, while fall is the worst. For the brushland habitat, the deer are in poor condition throughout the year, but they are in a little better shape in spring and poorest in early winter. One interesting conclusion in this study was in regard to the deer-parasite relationship. Instead of finding that animals in poor condition had the highest parasite counts, for instance of abomasal worms, the deer in each area had a characteristic level of parasite numbers, regardless of their condition. For instance, the redwood deer averaged over 850 abomasal worms, the woodland prairie deer only 50, and the brushland does an intermediate 250 worms per deer. Other parasites, such as Sarcocystis, footworm, and tapeworm larvae, were a function of age. The older animals had higher counts than young animals. The intermediate stage of the tapeworm, Taenia hydatogenia, was commonly found as a cystocercus on the liver. In the adult stage, it is a common tapeworm of the dog family, but it is harmless to both the deer and the canid. When found in the mesenteries, it is called the bladder worm. When the deer is eaten by a coyote or dog, the tapeworm scolex inside the bladder attaches to the canid's intestines and matures. This tapeworm does not grow in man. The cystocerci are also often found inside the liver. The intestinal worm Esophagostomum was found in low levels in 11 of 28 deer, and lungworm Dictyocollis occurred in 5 deer. Both occurred in all three habitats. Such parasites as the eyeworm, Trichuris, an intestinal parasite, and arterial worms were found only sporadically. The presence of these parasites did not seem to correlate to habitat, age, or other factors. Because of manpower and funding limitations, this study was not broad enough to investigate all factors influencing the deer. Some analyses, such as of rumen contents, habitat conditions, and the developing of growth curves for fetuses, will have to be done as separate studies at a later date. A sample of the rumen contents of each animal was taken and stored in formalin for a future time when the partially digested food could be analyzed for species content and seasonal food preference. The dissection of the head, removal of the jaw, and the search for parasites in all parts of the head were done at one time. No evidence was found of ear mites or meningeal worms, Paralaphus strongylus. These have been commonly found in other deer herds. The arterial worm, Eleophora, is found in the arteries of the head and neck. It was found only in deer from the brushlands. None were found in this animal. After removal, the jaw was used to age the animal by checking for tooth eruption and by examining the extent of tooth wear. Thin cross-sections of teeth were also made, and the annual rings in the cementum layer were counted as another method of age determination. This jaw, from a very old doe, about 12 years of age, shows considerable wear in the incisors, premolars, and molars. By comparison, the teeth of a yearling show sharp ridges and crowns. One more member of the normal parasite load, which was found more frequently in older animals but is harmless, was the larvae of the botfly. This maggot resides in the nasal passages and sinuses and feeds on mucus and other tissue products. It is deposited on the nostril by the adult fly and crawls into the nasal cavity where it matures, then crawls out and drops to the ground to pupate. Body fat measures, carcass weights, and blood values were studied for their potential as measures of the condition of a herd. Because most of the deer had very little body fat, it was found that the blood values and carcass weights were the best measures of the actual physical condition of these deer. Here the hematocrit, or packed cell volume, is being determined by centrifuging to separate the blood cells from the serum. The percentage of cells is then read directly from a hematocrit scale. It was found, however, that neither the blood values nor any of the condition measurements correlated well with the reproductive data. A high number of red cells per cubic millimeter indicates good health, but a high white cell count suggests that the animal is undergoing some stress. By doing a differential count, the quantity of each kind of white cell present can be determined. A high number for a specific kind of white cell can indicate either a bacterial infection, allergy, or heavy internal parasite load. The one value that correlated best with the actual condition of a deer was the hemoglobin content, as determined with the use of a hemoglobinometer. In future studies on Humboldt County deer in which no information on reproduction or parasites is wanted, a sample of blood could be all that is needed. A smear of blood on a slide is placed inside the instrument and simply compared to a standardized colorimetric scale. This study has provided a large amount of information about Humboldt County deer herds. Although it has revealed the current status of these deer, its full significance is more far-reaching. It has provided baseline data against which the figures from future studies can be compared. This will provide an accurate way to judge future changes and trends of deer in Humboldt County.